happens. Okay.
Okay.
Great. Take it away, Federico.
Okay, thank you, Matt. Thank you for the introduction, and thank you for joining us, and also thank you, Annekathrin, also joining me in giving this workshop to you. I'll just share my screen as a starter. As Matt said, you can feel free to post questions in the Slack, but also very much, please do interrupt us while we present. I mean, there is not too much of a hurry, and I really do welcome questions in between. And if you're comfortable with that, you can open up your microphone. Otherwise, type it in the chat. We try to capture those.
でしょう。すみません。
はい、カズ。
もしもし、すみません。ヘッドホンをつけます。聞こえますでしょうか？
Sorry, say it again.
As far as I heard, it was Japanese, so maybe.
Okay.
Even not for us to understand.
Oh, okay. So my language skills is up to R on that side, so I cannot get that. Okay, let me just start the screen, the presentation for this, and welcome everyone to the GeneTonic Workshop, or the package demo, where you can probably really try to enjoy the interpretation of RNA sequencing data analysis. It's a big pleasure for me to join you at BioC Asia. That's my first time there, so there. You should have some information about the workshop. Of course, it could have been already passed to you in advance. So there is a fully fledged package for that. And that package is pretty much what is used to fire up the orchestra instance that you can use if you want to reproduce these things on your own without having to install things. So you can find us on Twitter. So I'm sometimes very active, sometimes less. I'm Fede Bioinfo, and Annekathrin is Annekathrin Ludt. You can reach to us also there. And we will try also to capture, to answer all of your questions also in that venue if you think that it can be helpful for you. So if you are here, I'm more or less sure that I don't have to tell you too much about what RNA sequencing is. Think of that as a nice, high-dimensional, very quantitative and sensitive snapshot of what goes on at the transcriptome level in cells or in single cells for many samples and for the different genes that you have at hand. So to boil it down, you end up having a large matrix, a fairly large matrix that is genes on the rows and samples on the columns. And these samples can be your milkshake of cells if you have a bulk RNA sequencing, or the single berries if you have a single cell RNA sequencing dataset. What are your objectives if you do so? Normally, or in many of the cases, you want to compare the abundances of this expression quantifications that you have for the different genes, and you want to discover the so-called differential expressed genes or gene signatures, some features that are somehow associated with the phenotypic differences that you can observe in these experiments. If you go at the single cell resolution, then of course you might have the possibility to do a little more refined things. And these things can be the identification of cell subpopulations. You can describe the processes as a developmental trajectory. You can study the heterogeneity in the transcriptome regulation and so on and so forth. And as for the general, context, you have a wide variety of applications that exist if you are doing RNA sequencing. I'm a big fan of this overview that we have from a paper from Annekathrin and colleagues that pretty much summarizes quite everything, say in the pre-single cell era, what you could do and what you probably should do pre-analysis, in the analysis, and the advanced analysis parts with RNA sequencing data. So I say that RNA sequencing can be fairly easy, but this is not just because of us, let's say. We have a lot of established tools that help you, for example, take care after you have an adequate number of replicates and sequencing depth in your reads, and you have your row reads that you quality control, you align that or quantify that so that you can have gene or transcript level expression quantification. You plug them into a differential expression framework, such as, for example, Lima or the ESEC2 or HR. And then what you want to do is then try to interpret that. And you do that with this so-called functional profiling, approaches. You can visualize that so that you can have a clear idea of what goes on. Sometimes a picture is really worth a thousand Excel sheets. And you can integrate these data sets with different approaches, especially if you have different layers of omics that you have coupled to this experiment. But in the basic scenario, what you do is that you have Bioconductor, which is a very nice framework that provides you not just one single toolbox, but a different set of toolboxes, because you can do quantification, for example, with star or salmon. After that, you have, for example, in the end, you might end up having something like a summarized experiment. So your expression quantification would go here, then some extra information on the genes would go here as a raw data, and the cold data would be the extra information on the samples. What you can do with that, you can, if you before asking a question or before answering something, you might need to ask the right question. And you can use, for example, the Explore Model Matrix package that does this and can help you through this by kind of giving you a loop into what the formula, so the model matrix is. You can use IC for simple visualization tasks or also refined ones. And one of the possibility to use after you have the data in a summarized experiment, you can use the ESEQ2, but you can also use EdgeR, you can also use Limma. And very nicely, these three formats are very much interoperable, thank you to the DE formats package. So if you want to use a package that wraps pretty much the ESEQ2 framework for that in an interactive way, in a similar way to what, for example, GeneTonic does, you can try out the ideal package that I've been developing, and that can help you generating the novel, a set of novel hypotheses and insights that you can use to actually extract knowledge out of your data. So in Bioconductor itself, there's a huge amount of resources that take you through the main workhorse for the RNA sequencing data analysis. And you can use these nice workflow packages, the RNA-seq Gene, RNA-seq Gene EdgeR-QL, RNA-seq 123, and RNA-seq DTU. These are very similar to each other, if you ask me. And then there's the DTU one, so for the differential transcript usage, which is a slightly more refined way of approaching this differential expression context, right? Not just on the gene, say overall level, but also on the differential transcript usage that can switch between conditions. What we have with Bioconductor is that we have efficient methods and efficient data structure. So everything is there, more or less. The pipelines are all set. Imagine that your bioinformatician colleague or your very colleague, which is not a bioinformatician, but really worked out his or her way through the pipeline, is there and just got a nice set of results. So what do we need now? What we need now, to me, especially in collaborative context and interdisciplinary context, like we are kind of blessed and forced to work within, we need a powerful framework that communicates these results. And in my experience, I found out that pretty much the combination of R Markdown and Shiny can really bring up to a lot of, say, love in terms of efficiency and ways to do things in a proper, yet, say, enjoyable way. So pretty much to the point that we are at the real bottleneck that prevents you to sparking joy in the analysis, we are actually there at the data interpretation. So Marie Kondo, as a spirit guide for that, tells you that you should probably reject or say escape table land if that's the painful way of doing it. So we should do something, the data interpretation in a way that tries to spark joy into your analysis. And if I have a couple of lessons that I've been learning throughout my say years of experience as a bioinformatician, is that it takes at least two, or right now, actually, I could distort this saying by saying it takes almost 3 to tango right now. You need results and interpretation, so just not the results that you generate, but you need to make sense out of that. You need at least a bioinformatician and at least an experimental scientist. Many times you also need, for example, a physician, a medical doctor, and so on and so forth. And you also need, or you could use very nicely, a combination of reproducibility and interactivity. So this is kind of the holy grail of this graphical user interface-based applications, that it's not just a point and click interface, but it's something that you can really nicely use in a way that somehow supports the reproducibility of your analysis. So how to do that? I've been there, I've done that. So it happens that I myself generate a lot of Excel tables or Excel-like tables. You shouldn't use Excel for bioinformatics. There's already quite a couple of papers around that suggest you to not do that. Otherwise, Sept1, your gene becomes the 1st of September. So Excel is pretty much like a dating app before Tinder to me. So because everything's a date in Excel. So imagine you have a lot of tables, and a lot of tables. You have the quantification, you have the differential expression analysis, you have the results from enrichment analysis, and you might want to have different, say, views on that. You might use different tools, and you might also need, for example, some tables to interconvert between the stable identifiers that you have to the HUGO, so to the gene symbols. How to do that? So it's been painful. It's been quite some pain doing that. So we try to answer this, or at least to propose you a solution, by putting everything into a glass, a cocktail glass, where all of these ingredients are shaken for you and pretty much mixed up in a seamless way. So basically your effort might become minimal. How? So why did we do that? It's a comprehensive toolkit that tries to streamline the interpretation of this whole functional enrichment analysis steps, putting them into context with all the information that you have from expression values and from the differential expression context. This was one of the sentences that we had in our manuscript that is still available on bioRxiv, and we can right now actually very happily say that it's been accepted last week. So soon to be impressed at BMC Bioinformatics. So watch out for that if you want. And what do we have? We have these 4 building blocks. And these 4 building blocks can be put together in an efficient way. The efficient way is that we enforce some kind of standardization of the objects that you use for that. So don't use Excel sheets, because Excel sheets are easy to modify and easy to mess up. We have this so-called one-stop shop solution that you can use after the steps that you commonly perform. You can have a lot of visual overviews on all of the enrichment results and the expression data. So it's not just the table from for example, from Cluster Profiler and its visualization, but it might be a little more information once you have also the expression matrix. You can have the beauty of having interactivity and use that to perform the so-called drill down operation, so that you can have a little more detail in what you see just by clicking on objects and observing that. As a result, you do reduce the time to insight to generate actually these so-called actionable insights, or some people like to bring the bench to bed. So from the experimental sides to the patient-related side, you can get better results and in a together way. Since it's Bioconductor, we wanted to have interoperability as a key design aspect. And these shakers are kind of the symbol for that. So think of it as GeneTonic actually does not give you the enrichment tools, or let's say, you do not perform enrichment analysis in GeneTonic. You feed the same set or the similar set of objects to GeneTonic, and these can come from different pipelines of yours. If you are familiar with Cluster Profiler, perfect. It's an excellent package. If you're fine with results generated by TopGeo or by GeoSeq, you can all use them and use these shaker functions that basically convert the output from these functions into something that can be readily fed into GeneTonic. And I don't know if you see that, but say right now here, there is a tiny bookmarking button. And this bookmarking functionality is what you will see just later. That's useful, for example, for reporting and for reproducibility. So in a Shiny live session, what you can do, you end up, say, being interested into different features, how to keep track of that. You can see that how it works in Annekathrin's demonstration. So the GeneTonic workflow, again, looks like something like this. So you have the DE-seq dataset. So we are not enforcing you to use the DE-seq framework. You can just convert the results, for example, from Lima or HR to these formats. But right now, so we take these as input. We do show, for example, in the supplements of the the manuscript how to come from other frameworks and use that. Then you have a data frame for the result of enrichment and an annotation object, which is nothing but the interconversion between gene IDs and gene names. Put them together in a way that fits into a so-called GeneTonic list. This is optional, but it makes things easy because it's not 4 objects that you have to keep track of, and it's just one list-like object. We will be formalizing this probably into an S4 proper class, just to make it even more robust. But what comes after that? So what's the deal? Once it is in GeneTonic, you can have a lot of interactive exploration for all of these objects. You can have a lot of visualizations and put together all the things that you do in these 2, say, meta functions into a nice integrated reporting. So whatever gene sets and whatever genes you focus on, you can just pretty much with a one click on a button on the so-called happy hour function. So it all fits in the naming scheme. You basically end up having an HTML report which is focused on the features that you selected. I think there might be a question, or did I? I'm hearing some clicks, but so I don't know if it was a question. Is there a question? I'm happy to take a quick break. Otherwise, I will go on.
There is a question in the Slack that asks, must the rows be genes? This is from Jeremy. I work with lipids, but have similar things like lipid set info, lipid statistics, lipid annotation. Thanks, Jeremy, for your question.
Yeah, yes, I used to say that when we want to talk to a statistician, we have to turn our head like that. So we have to tilt by 90 degrees. It doesn't have to be genes. So probably, okay, I think I mistook the question. So genes or lipid, it can be lipid identifiers per se. It can be lipid DE, so it's not DE genes, but it's DE lipids. As long as you can somehow then interconvert to the, say, GO or other frameworks where you actually have lists of such features that are annotated to this overarching functionality. I never work with that, but I would be more than curious to see how it works. So if you want to reach out anytime, I'm interested in giving it a look. Otherwise, probably the question was not about, say, the orientation of the table, but probably just if it fits with, say, lipidomics data sets, I guess. We never tried, I guess. Probably it's possible, but I don't want to put my hands in fire if for that. I'm not so sure that there is some kind of most direct way of going from lipids to GO, for example, or to M6DP signatures. If there's no other question, I just would go on and then probably just say try to collect them afterwards. But say, yes, the whole point is that the shakers basically pretty much can help you bring the output of many tools that you use for function enrichment in a pretty much seamless way. So if you use David, if you use Cluster Profiler, FGSEA from Bioconductor, TopGO from Bioconductor, GProfiler, Enricher, all of them are currently supported. If you have any other tool that you want to be supported, then just reach out. Either I will be happy to make to implement kind of a conversion function for that, or if you want yourself to contribute to that with a pull request, we heartily welcome that. So if you want to try that out, the local version of doing this would be with the classical BioC manager install function, and provided that you install BioC manager, it's basically one click to say one call to say install GeneTonic. And if you want to install the development version that is on GitHub, so that's pretty much the debel that you would get if you use the development version of Bioconductor itself, you can do that like that. If you want to install this workshop has GeneTonic plus a couple of other dependencies, and you might want to build the vignettes if you want to have them available locally. But say this is actually not to be done because the package has been rendered already online, and you might be all set to have your first GeneTonic. So in this quick package demo, we will show the data set that we've been working with, in a paper from Alma, in a colleague of mine. She had a very simple, actually, data set with two conditions. In one, she had triplicates, and the other one she had duplicates. So it's a fairly simple data set, but it's easy to see, for example, how many of these nice-looking plots you can generate just by pointing and clicking the mouse. And you can otherwise use the macrophage data set in the package that is available as an example in the package. One of the problems that I thought, at least, in the moment where I have a very rich user interface, is that you can really overwhelm the user, and you don't want that. So we didn't want that. We wanted you to actually have one way of learning by doing in GeneTonic, and you will see a quick demonstration for that with the so-called tour functionality. Tour functionality you can consider that as a non-static way of documenting how to use a tool, how to use a graphical user interface. You will see how a spotlight basically goes around and shows the different elements of the user interface, how to use them, gives you suggestion on how to use them. And that's neat magic is implemented with the intro.js package, which is wrapped by the r-intro.js R package. And yes, as I said, a picture is very much worth 1,000 row tables with 12 columns or more. So you can have a volcano plots of your gene sets. You can have a representation of the individual components for each gene set to see, for example, how the logfold change of the different members behave. You can have a summarization as a Z value over the samples for the different pathways or gene set that you have. And you can display that a gene set by samples, say heat map. Otherwise, you can also, for example, compute some kind of degree of similarity or overlap between these, and you can have an MDS plot for that. And once you have that, you can, for example, encode some additional information, such as the Z-score that tries to give you a kind of an idea whether that function is going up or down, just by summarizing that as at the gene set level. So I think I've been showing you a little these links already, but just to collect them in one place. As I said, GeneTonic has the package on Bioconductor and as well on GitHub. Then the GeneTonic Workshop repo is there. The manuscript is available on bioRxiv and soon at BMC Bioinformatics. And if you want, actually, I can give you an appetizer that we are writing a series of protocols of how to use, for example, PCA Explorer, Ideal, and GeneTonic to have a consistent framework where you have this combination of interactivity and reproducibility throughout all the phases of differential expression analysis, from exploration to differential expression analysis downward, all the way down to interpretation. And of course, you can feel free to reach out if you have any suggestions and contributions. And with this, I think I'm pretty done, at least with the introduction to that. So I'm gonna switch back to the main, to the other tabs. I think you are familiar right now, you might be familiar right now with the orchestra system. So I'm gonna paste this in the chat if it works. Let me see whether this works for you. Chat. The chat is, okay. I think I've seen that the chat was not active for the.
Yeah, probably you have to put it into the Slack. Maybe that works better. Yeah.
I'm just gonna put it here. It should be also in the pin messages, so but just for the sake of making sure, make sure that once you are ready to launch the workshop, you can use the RStudio username and password to log in. And if you do so, then you have such an environment where the whole package, so the GeneTonic Workshop package, is available for you, and you can just run it from there. And with this, I think I'm gonna pass it over to Annekathrin, unless there are questions. I mean, in the meanwhile, Annekathrin can probably start sharing the screen so that we don't have too much of a idle time, but we do welcome any question. I'm also checking the Slack for that. Just in case genes come out afterwards, questions come afterwards, do write them here and we will make sure to pick them up.
So now you should see my screen, right? You should see
the
vignette.
Yeah, perfect.
Perfect.
Okay, I think if there are no questions, you can just start, and then maybe we can take questions as you present. I'll make sure to check out also the chat and the Slack. Okay.
Okay, yes, that would be perfect. So good afternoon, everyone. Also from my side, I will quickly show you the vignette to this workshop and how you generate the objects for GeneTonic. Probably you're all very familiar with how to generate your RNA-seq data and later on give them to GeneTonic, but just in case you're a little unsure, we just want to show you, and afterwards I will dive right into one instance of GeneTonic and just show you a little bit around. Yes, so we start with the setup for this workshop. As Federico already showed you, you can install GeneTonic with Biosc Manager, you can install the develop version and the workshop. And after you installed it, you can just load it with the usual library command, or you can use Orchestra where it has already been loaded. Today, we're using GeneTonic on this A20 deficient microglia dataset, which is a very small dataset, but still good enough to show you how much you can already do with GeneTonic on a smaller dataset and how much you can see on your data with the different visualizations. We start by loading some packages, and we already provide you with the data in the repository, so you don't actually have to prepare the data. You can just install the workshop and you're good to go. You can directly start because we already provide you with this RDS objects. But I just want to quickly show you how to generate them so that you know for maybe your own data or for further usages of GeneTonic. We start from a DE-seq data object, which is this use case DDS ALMA RDS. We load this, just have a quick look at it, do some transformation on it, and then look at the small heat map. And we see here we have like these 3 condition samples. We have 2 control samples. We see similarity between the control samples, between the treatment samples. One seems to be a bit off. And we also have some PCA Explorer, some PCA here from PCA Explorer. And here again, we see some nice separation between treated samples and control samples. Again, this treated sample R1 seems to be a little bit off, but nothing too major. But maybe we can keep it in mind later when we look at GeneTonic. So also it could be something you could do with your own data, just do a PCA beforehand, before you go into GeneTonic, and maybe you see something you want to have a look at with GeneTonic. You can do that very much. Next, we do some differential expression analysis to provide the second input of GeneTonic, the DE-seq result object, which we just use the usual DE-seq workflow, re-extract the results treated against control, do some shrinkage, prepare some annotation data. That's more like for us to convert the identifiers from, in this case, I think it's ensemble, to some more human readable identifiers, and have some summary on our DE results. And basically with that, we have our second input after we run the DE-seq workflow. But you can also, as said, just basically load it from the package because we have already provide you with the final object you just have to load. We quickly included here some MA plot. You see we have many upregulated genes, some downregulated genes. So you just have a quick overview about how the situation looks like in this data set. We also provide you a little table about which genes are DE. Maybe you want to have a look at it after the workshop. Next, we do some functional enrichment analysis, which provides us with the third input to GeneTonic. In this case, we use the top-go package for our functional enrichment, but as Federico already told you, you're not limited to this package. We provide also the possibility that you use other packages for your functional enrichment, and then you can use our shaker functions to shake your results into the right format so that you can directly input them into GeneTonic. In our case here, we're using the biological process ontology from Go, but you can also use the other two. We provide you here with the code how to use them. Then we also shake our results into the right format, and we compute some aggregation scores, which just have more like a visual purpose, so we can nicely color our visualizations later by these scores. Here we have a quick tabular-like overview of the processes, but also, again, you can just simply load the object. You don't have to generate it, but above you have the code how to do it. And then here we have
pretty much the, sorry to interrupt, that was pretty much the, say, the necessary set of information that we kind of request from you to have. It's nothing but an identifier, a description for that, a significance level somehow that you would obtain anyway from a test. And what's best to have is also, for example, the GO to genes. So this kind of relationship, what genes have been associated to that particular term, because that is actually what is used to, for example, to display the nice interactive graph that Annekathrin will show you later. Okay, sorry.
Yes, no problem. So here we also have some little paragraph about how, when you want to use alternative enrichment analysis methods, how you can use those. We provide you some little code and how to, again, shake them into the right format, so you can have a look at it if you maybe want to use a different package for that. And then we are actually good to go and can assemble our so-called GTL, our GeneTonic list object. That's basically just a list of the 4 different inputs you need for GeneTonic. It's a little more, let's say, a little easier if you have it in a list, because you just have to provide GeneTonic with one object, but of course you can also use the 4 different objects individually and input them into GeneTonic. Yeah, but we think it's kind of convenient to do it in this like list object, have one object you need to save, and then you're good to go. Again, this GeneTonic list object, you can also load it. You don't have to generate it yourself. And then we can directly start with GeneTonic and start our GeneTonic instance with this call to the main function and providing our GeneTonic list object. And now I think I need to stop sharing to be able to open up GeneTonic again. So now you should see an instance of GeneTonic, right?
Yep.
Yes, okay, perfect. So I just did what I showed you there, started the instance with the call to this GeneTonic function and the GeneTonic list. We are here in the first panel you will see upon launching GeneTonic, which is our welcome panel. You see your 4 different inputs. Here on the side, you have some nice little summaries, but you can also click on these plus signs in these individual boxes and have some, little more detailed look on your data, some little sneak peek. You can look into your data, you can click around and just have a look. Let's see, here we have the DE results. We can also see here a little colored, the log 2-fold change for the individual genes, p-value. So here you can just have a look at your data, the functional analysis results and the annotation info. So here you basically just have a short little overview of your data. And then we can directly dive into GeneTonic by changing here over this panel selection on the side. We can select the second panel, which is the GeneSet Panel. You see it loads for a little while, this GeneSet graph. But what you probably want to do first when you start using GeneTonic, you probably want to have a look at the tour. So because the tour will simply explain to you how to use GeneTonic, which information you have in each panel, what you can do in each panel and in each section, and just in principle will guide you through the whole app. So let's have a look at this first. You can start a tour by clicking this little question mark button. You will usually find in the upper right corner somewhere in each panel. So we click it and you see that this little tour opens up, which gives you a nice little welcome to GeneTonic, tells you in which panel you are. And then with this next and back buttons, you can basically click through the tour. So we look at the next element. It tells you, yeah, you can move around here. And you see that it will highlight the individual parts of the panel it explains to you so that you know which part of the panel the tour is currently talking about. Now you have some information about the main content of this tab. You see that this tab mainly displays a bipartite graph of genes and gene sets with the aim of making the exploration of the relationships easier. Then we can again click through it. We see here we have this graph. We need to scroll a little so that we see the tour again. The main, it says, yeah, this is the main network which will be displayed. You can change the number of nodes and so on. And we can click on. It says we have some little control bar here. We can toggle. And you can either click through the tour completely, also selecting individual elements of the tour by hovering over this little point, or you can also close the tour if you have seen everything you want to see and just get back to the panel. So now we have some little impression about what this tab will do. It will, as it is called, display you some relationship between the genes and the gene sets in your data. And this yellow rectangles in this graph are your gene sets, while this oval shaped little elements are individual genes. And when you see it, when I hover over this graph, you see that either once this little selection button changed and also individual elements are highlighted. So we can also click on one of these elements. Let's select this gene set here. And you see that here on the right, this gene set box will open or will pop up when I select the gene set. And here it opens up some further visualizations. We have some signature heat map in the individual samples of this gene set. And we also have a volcano plot with showing the significant genes from this gene set and also the background distribution of all genes in the data. And we also have some further information here. This is some little clickable button. You can click and it will directly bring you to further information about this specific gene set. It gives you here the term, the name of the gene set, some p-value information and some definition. And we can also do, so if you click on the gray background and the selection will, you deselect the gene set and we can now, for example, select a gene. For example, let's take this one. And then we see below this gene set box, we have a so-called gene box, with some further information about the individual genes. Here we see that we have CCL5 here, and we have also some clickable buttons with links to different databases, for example, the NCBI gene database. So you can just click these buttons, get more information about the genes. For this gene, maybe it was not a good one I picked here. We don't have an adjusted p-value or a log for change. So maybe we select a different one. Maybe let's take this one. Okay.
I noticed that that was not also showing as a labels in the volcano plot somehow. I don't know why. So imagine that, say, if everything runs fine, then you will also have the statistics from the DE over there, and then the annotation from there. What you can do maybe is that if you highlight any, say, for example, the condition from the control bar on the right.
Yes, I want to do that next. So you see here, it tells you, please select a group for the experimental condition. And to select this group, you click on, you toggle this icon here, and then you can select here group color by. And here let's take condition, because we have seen that our data groups by condition. We close this sidebar, and then we see that we have a little plot here about the individual group by condition. We see the experimental group, control, we see the treatment group, and just can open up some further nice plot. But let's have a look again at this little sidebar. What you can see here, you can, for example, change the false discovery rate for DE. You can either do it by using these arrows on the side, or you can also yeah, just mark it and use your keyboard to change the number. And you can also change the number of gene sets displayed in the graph. So maybe we change this one. Let's take, for example, 30, which is just doubled. And then you see this graph up here will again load. And yeah, this might take a little while. Let's wait for it. And now you see that the graph has changed. We see a little more gene sets. And we, for example, now also see some downregulated genes here. So maybe let's take this one. Okay, something seems a little off that we don't have p-values. Maybe we should have a look at it. And yeah, here we nicely see downregulated genes. It's upregulated in the control samples. Or we have more counts in our control samples, less in our treated samples. And yeah, basically, so you can, with this, you can click through this GeneSet graph. Some nice new functionality we just recently added is this little button down here on the left. If you hover over it, it show the code for this plot. So you can click it, and then it will open up a little code section showing you how to generate this plot with code from GeneTonic. So maybe you want to replicate this, this plot and want to include it in your paper, your presentation, or something else. And here you just simply have the plot, simply have the code to generate this plot, or you can also save the GGS graph by clicking here this button. Furthermore, what Federico already also told you, again, we have this nice bookmarking functionality. So maybe you see a gene set in your data, which is really interesting. You've found some interesting information about your data, about your gene sets, and you want to save that for later because you don't want to search for it later on in a second session, or it's just that gene set you were looking for. And
now the labels are there. Okay, I'm puzzled, but yeah, okay, let's just take it as it is. Maybe you can try to zoom in a little in the graph so what I hate of these graphs is that they are too large, too much hairball-y. But when you do basically zoom in, these graphs become usable and alive, right? So that's, I'm back in love with graphs now.
Yeah, you can also, when you zoom in, you see the names of the gene sets or the genes displayed in the individual elements of the graph. Of course, it's hard to see if you're zoomed out, but just you can zoom in. So again, let's say maybe this is a gene set we are particularly interested in, and we want to save that for later, or maybe export information about that. And that we can do by bookmarking it. So we select the gene set and then click on this bookmark button. And it tells us again here down here, edit antigen processing to the bookmarks. You can also hit the left control key. I think it's the left control key. I usually use the mouse to bookmark genes and gene sets. So if you rather use your keyboard, you can do that. And maybe
basically that was lazy me not having to travel with the mouse to the bookmark panel every time. You just kind of hover over things that you might be interested in and left control, left control, left control, like on a special sale online. You just keep on adding to your basket items of interest.
That's so it's pretty convenient also if you're maybe you're down somewhere in the graph and you don't want to travel, just use the keyboard. Let's, for example, now also select a gene. Let's take this one. We can also bookmark this gene. It tells us edit to the bookmark genes. And if we now go to this bookmarks panel here on the left, navigated with this panels to the bookmarks panel, we see that we have one bookmarked gene, the one we selected, and we have one bookmarked gene set, also the one selected. And now you can generate a report, including these the bookmarked genes and gene sets, some information, some visualizations, with clicking this start the happy hour button. I will not do it right now because it takes some time to generate the report, but you can just probably have a look afterwards and just test it out. You can also export your data as serialized summarized experiment. For example, if you want to explore your data further with the IC package, you can do that and directly input then the data to IC. So this is like our nice overview for reporting, bookmarking, and reproducibility you want to use. And I don't know, maybe we want to go over to some questions, or I just keep showing a little more on GeneTonic. We have about 10 minutes left. What do you think?
I can say, for example, that the start the epi hour function will probably not work on the orchestra because it might be that you don't have the permissions to use it there. But if you run it on a local machine, that should just work out of the box. I don't see any particular questions. So maybe, I don't know, okay, there is a question from Jeremy. Is the GeneTonic able to save the data processing process if I refresh the screen or leave it for too long, or will it restart and I need to resubmit my data again? And I think, so the session, the fact that Shiny kills your session if it's unused for too long, that's pretty much something inconvenient. You could, for example, have it on a stable non-server-like machine. So if you have it locally, then it doesn't kill the app. There is no functionality per se to save, but you can still, for example, you can use these vectors of bookmark genes and bookmark gene sets just afterwards to have it. That's actually a cool feature that we can do, that we can pass the bookmarks again. I just note it down into the app poll. And then we can, for example, have these, say, persistent storage of these elements. That's a very good point.
Yes.
And there's another one. Is there a way to filter the graph so that it has less nodes and edges to look at? Good point. Not in GeneTonic per se, but all of the objects that, for example, the GeneSet graph do return you are all iGraph objects. So in principle, you can generate them with the functions that you have. So it can also work outside of the app. Of course, it's a convenient thing to do with the app. But in case you want to use them as, say, in your scripted analysis, you can also do that very nicely. Yeah. And there are some ways that maybe we can show later this function to kind of reduce the redundancy of the terms. There's another question from Matthew Ricci. So whether in the latest Bioconductor release, whether we had any major changes or newest features that we added to GeneTonic that we would like to highlight. That's an excellent point. I think one of them was already showed by Annekathrin, and that was actually a nice point from reviewer 4. That's about, say, meta-generating the code for that. It's not the holy grail for automatically generating everything, but it's, for example, it's a nice didactical tool that we have for that. We have in mind one feature that we can pass and that we can implement. And if it's, of course, more visualization, this will come because we might have some kind of more complex structures and complex relationship that we want to display with in nice and colorful, not table-y ways. But also the fact that we can, maybe related to the fact of sessions, like we can also, for example, run GeneTonic as a kind of a server where you can upload this object, this GeneTonic list as an object, and then basically GeneTonic loads automatically for you. So pretty much you don't even need to think of having to type read RDS if that's not your, say, this is not your skill set, because sometimes you just want to upload an object and explore it. This is something that I expect to happen soon-ish. Say, it will need some kind of major reworking of the whole API, but so we might be up for it. And I see there's a question coming. What file type does GGS graph say the graph is? As I said, it's all iGraph objects. I think maybe you can display that, Anka, if you click on the code, on the code generating button. You should be able to see this G object is nothing but this iGraph object. That iGraph object gets then passed to this network, and that pretty much does the magic of rendering it nicely. Because say you kind of probably know how not so beautiful are the standard plots of iGraph. Basically, all the nice visualization features are handled for you automatically in the GGS graph function. So there's quite some code, mouthful of code going on there, and you don't need to see that, but you can see that if you want to see that. You just need to use the source. And can I reload the graph to another graph to like Cytoscape? Yes, I wanted to, I think Cytoscape could accept, I don't know if Cytoscape does accept iGraph objects as say per se, but maybe something like the CIF, the simple interaction format as an encoding, this should be fairly easy to convert an iGraph object to a CIF object. And the same goes, for example, if we want from iGraph to, we will need to check out Cytoscape requirements that you would need to pass for importing. That's another good point. Thank you, Jeremy. And yeah, in the meanwhile, maybe, I don't know if you want to just show off the enrichment map feature.
Yes, I can do that.
And we increase the number of gene sets, maybe. Let's say, imagine that you have a 200 rows table, that's or 300, 200 rows table with the results of enrichment. The enrichment map is, to me, a very nice feature that can try to give you some kind of a biological overarching themes. And you can, for example, by coloring that by the Z-score, we can see, for example, how for example, these terms are actually do cluster together, for example, depending whether they are going up or down. That's kind of not unexpected. And if you want that, so in the end, if you have a graph object like that, you can do a kind of a distillation of this in a more quantitative way with the so-called GeneSet distillery. So we wanted to stick to the theme of beverages. And this does generate some kind of this distill EMAP, for example, button down below. And you have, so now it's back to 50. So we could probably have to say 200 also in the box below, if I remember correctly. So if you take that, then it basically takes the graph and then tries to display the similar graph. And you can select these so-called metagenes, which are clusters of nodes from the graph object. And if you click on any of these, then you obtain a so-called metagene set box. And we display a gene set which is representative for that cluster, but tries to kind of compact these nodes together. So it's not the answer to the how do we display a more polished graph object, but it's one way to try and compress this information. Thank you, Annekathrin. It's always nice to see my voice over. We don't have a voice over functionality to actually operate with GeneTonic yet. Yeah. If you want to check that out, it's a functionality that we had in IC, and it was quite fun to implement. It's I don't know. I'm still thinking how crazy it was to actually think of that, and how fun it was to implement it. But that's a different thing. Okay. Yes, probably another question is coming. I see someone is typing. Also from other attendees. I mean, we do welcome any question. Otherwise, we are approaching the o'clock time. I see also Matt.
Feel free to raise your hand if you want to ask your question live. We can unmute you, or you can probably unmute yourself.
does the size of the node in enrichment map mean anything? Yes, it does. Maybe you want to take that, Annekathrin?
It's the number of genes in this gene set, as far as I know.
Yeah. So it's encoding the gene set size. So
yes.
A smaller one is a more specific term, and a larger one is a very general one. If you, this is something that happens, for example, if your enrichment framework returns you two general terms, then you might have your big companion. You have a biological process that eclipses pretty much all the other nodes. But I'm pretty sure that you don't want to have biological process to pop up as one of your enrichment results. So GeoSeq, TopGeo are very nice, for example, examples of algorithms that return you either RNA-seq specific worked out gene sets and decorrelated gene sets that basically try to aim to tell you what goes on at a more specific level instead of just giving you too much generic terms.
Yes, you can also see like the size of the gene set. Now I'm hovering over some larger circle. You can see here on the side in the gene set box, there are like in the heat map, there are many genes. And if I now hover over some smaller one right here, oops. So you see it's like 4 genes. So the heat map can see a little more in the heat map. Some more specific term compared to the one before.
Yeah. There's a comment from Stephen, but say thank you for the nice words. So feel free to play around with it in the next few weeks. Yeah, I mean, we didn't show you the whole generation of this visualization. It was just in the other tabs. You can just kind of feel free to click and then expect Shiny to just take a little while to load all of these visualizations, and that's going to be it. Otherwise, I think we can try and wrap it here.
Yeah, any last minute questions?
Yeah.
If you have any over the coming little while, feel free to add them to the Slack, and I'm sure Federico or Annekathrin will be happy to answer them.
Yeah.
But if there are nothing further coming through, I'd like to thank both of our presenters for a really great happy hour this afternoon that we've enjoyed this evening. I think we've all enjoyed our GeneTonic today, and yeah, encourage everyone to explore it further on their own data over the coming period. And yeah, it looks like a wonderful tool. Thanks both.
Thanks.
Yeah,
from one of the Glimmer creators, it means a lot. So I'm a big fan of Glimmer as well. So, and I like a lot so how we are actually now moving towards making it easy to interpret the data as well, and not just generating set of results, because that's fairly easy. Where it becomes time consuming is the interpretation, and we are finally paying an extra eye to that. So I think our collaborator friends will be very thankful for that.
I totally agree. Anything that makes it easier for them to have a look over their data and makes it more fun for them to do that is a good thing, as opposed to just spreadsheets.
And it makes us lazy or say efficient somehow. That's the kind of the benefit that comes as a side effect.
Well, thanks everyone for attending, and thanks again, Federico and Annekathrin.
Thank you for having us.
Catch you later.